is, an, is in the Department of Biotechnology at here, and he's also a core member with the Robert Bosch Center and the IBSc, the Initiative for Biological Systems Engineering. He has received his PhD from IIT Bangalore. It involved the computational analysis of metabolic networks and protein-protein interactions in Mycobacterium tuberculosis for the prediction of potential drug targets. Subsequently, he did a postdoc at ETH, University of Zurich, Switzerland, involved the analysis of complex space of signaling circuits in yeast as well as synthetic logic circuits for their robustness and evolvability. His research interests are in computational systems biology and synthetic biology. Some pertinent research problems of interest are, specific problems are reconstruction of complex biological networks from data on metabolites, RNA transcripts, microarrays, and proteins, and the integration of these data into biological network models. SLAB is currently working on modeling analysis of metabolic networks for applications in metabolic engineering. So now let's take a deep dive into the undersea and zoom off to space.
Hey, thanks a lot, Sridhar. I don't know, you guys have dug out this introduction from really deep corners of the web. I don't know, Gita and I are wondering where you found all this information from. Anyway, so to get started, thanks again for this. It's really nice to speak to this audience, and I've really not got a chance to speak to this audience, although this is a very familiar venue for me. So today I'm going to talk to you about social networking in microbes from deep sea to outer space. But before that, a quick overview of what we do in our lab. So we are an interdisciplinary group focused on developing computational tools and scalable algorithms for a variety of applications, mostly for understanding and manipulating complex biological networks. And our major focus is on microbiomes and also communities from metabolic engineering. So if you see this, these are some of the tools that we use: network biology, machine learning, constraint-based modeling, high-performance computing, which plug into various applications. Could be metabolic engineering, biological data analysis, integrating multi-omic data. And today I'm going to focus mostly on this. And this is stuff I do with chemical engineering folks. I'm not going to go into this today. But this is something that's a big thrust in our lab, and lots of my students are working on related problems. So, and our ultimate goal is to have some sort of impact on the clinic or industry or environment. And yeah, we are very much part of the Robert Bosch Center and IBSc. These are some of the key research contributions over the years. In terms of algorithms and theory, so some of those that I've highlighted are what I'm going to talk about today. And also a bit of omic data analysis and so on, but I'm not going to go into that today. Today we'll focus mostly on how we've been modeling the microbiome using networks. So what is microbiome systems biology? Microbes thrive in a multi-species and dynamic environment. And so you find them everywhere, and places they are meant to be, places where we don't want them to be, and sometimes we want to eliminate them. But microbes, contrary to popular belief, they are probably more helpful to humans than otherwise. Okay, and they have a community structure that's dictated by a pretty much complex web of interactions, where metabolic interactions typically predominate, and which is why I'm going to use that lens to look at cells today. And there are many microbiomes of great interest to us, so it could be soil, gut, skin, and so on. And these microbial consortia are also very important in applications such as metabolic engineering. Not going to really go into that today, but my point is we need to understand how these microbes interact with one another if we are to, first of all, understand these microbiomes and try to see how we can, say, manipulate them and so on. So how do we understand the microbial interactions in these communities? It could be the gut, skin, or even the international space system. I'm going to go into many of these examples. And can we use metabolic models? Metabolic models have been heavily used in the past for various applications. And this is what a metabolic network looks like. So I tell the students this is like a very simplified view of what happens within the cell. This is already intimidating, but remember that each molecule that you see here has like 10 to the 17 molecules or something inside the cell. And there's just so much of complexity, so many pathways, so much crisscross traffic that you see inside the cell. But you can build these models fairly easily. Finally, to a modeler, we abstract the cell out as a long list of reactions of like A plus B giving C plus D. And how do we work with those? So this is finally my network, and I need to put a few networks like this together to get a community of organisms and try to understand what's happening in that community. And these are some classic, this is a very classic picture from almost a decade ago, where they show so many different applications of these, of just an E. coli model. So the E. coli model has been used for interspecies interactions, very little at that point of time, for metabolic engineering, for so many other applications. But today I'm going to talk about these interspecies interactions. So how to analyze these models? There are multiple ways to do this. So one is something known as constraint-based modeling. Again, something I've talked a lot about. I've spoken a lot about in different fora. But today I'm going to focus on simple graph or network-based modeling. Essentially like pathfinding in metabolic networks. And we can also predict new pathways and so on. But today I'm going to focus on pathfinding in metabolic networks to see how to understand how microbes cooperate with one another. And the important point is we need methods that are scalable because we are going to quickly go to large networks, large numbers of organisms and so on. So how to model microbial communities? You can again use the same methods that have been used for modeling single organisms, essentially constraint-based models, which translates to quite a bit of linear programming, quadratic programming and so on. So for those of you who want to discuss with me offline, graph-based methods, which I'm going to focus on, and there are also population agent-based models and so on. There are many challenges. There are these draft reconstructions, so there are important machine learning challenges in trying to make better reconstructions and so on. So this is something we've just started working on. And there are also an issue of standards in the community, because you typically have models that don't talk to each other, and you have two models developed by very different groups. To put them together and make sense becomes challenging. But nevertheless, there are many interesting applications that we find. Well, the question is simple. How do you convert a metabolic network to a graph? So Geeta was talking about road networks. So his junctions where multiple roads meet would be a node, and the roads themselves would be the edges and so on. So in this case, you have metabolites that are going to be the nodes and the reactions that are going to form the edges. So this is something known as a substrate graph. So you have these metabolites. So this is glucose. This becomes glucose 6-phosphate, which becomes fructose 6-phosphate and so on. But there is a challenge with this representation in that for this reaction to happen, it also requires a few more things. So for example, glucose plus ATP will give you glucose 6-phosphate plus ADP. So there is one more layer of complexity that's involved. That's a reaction graph which is not really so useful, so I'm going to just skip that. But this is a very, very useful representation which says glucose plus ATP goes into this reaction node, and out come glucose 6-phosphate plus ADP. And so on. So this is very similar to what you would see in a textbook. It will list multiple reactions, an enzyme, and out come the products and so on. But this is a bipartite graph. And what we want to do is pathfinding or something in this graph, but which is essentially a BFS, breadth first search. But the challenge is that you need to account for the availability of both reactants and things like that. You cannot just use a vanilla BFS. It's not going to serve your purpose. And there are issues with this currency metabolites and so on. So this is so there are these metabolites like ATP and ADP that are present everywhere in the cell, confounding your predictions and so on. So one way is to eliminate them, but in this particular paradigm, we are able to solve this problem without actually having to eliminate them as well. So how do we identify microbial interactions in communities? So this is where my first PhD student Aarti developed this very nice algorithm called MetQuest with a lot of help from Meghna, because without the efficient data structures, we wouldn't have been able to implement any of these algorithms. So this uses a bipartite representation of metabolic networks. What we use is a dynamic programming based enumeration, which assembling all the reactions that are happening into pathways of a specified size. So what are all the ways in which you can make a bunch of metabolites within the cell by mixing and matching different reactions? And there are two steps to this. We employed a guided breadth first search, essentially a breadth first search, but looking to ensure that all the reactants required for a reaction to fire are available. Otherwise, those reactions become stuck. I'll come back to that in a moment. And then assemble these into pathways. So you can just, this was developed for Python 3. What are the key features? It requires only the topology of the reaction network, no stoichiometry or atom mapping, which is typically required in some algorithms, making it harder. And it is simple and scalable to large networks. And invariably in graph algorithms, a pain point is cycles and so on. And in metabolic networks, there are obviously a lot of cycles, but we had an efficient way of handling them. And we also examined alternate routes for converting different molecules to each other and so on. And how do we handle community metabolic networks? This is about putting two networks together. So it's like, how do you combine two bipartite graphs? The idea is simple. So you have this one organism followed, and here is the second organism, and there are common metabolites that are exchanged. So there is F in this organism, so it's labeled F1 here, and it becomes F common, so to speak, and is then transported into the second organism as F2. So you can imagine that there are metabolites. Let's say there is a particular metabolite here that is not present in this organism. So when it's transported, it enables some more metabolism in that organism. So this is like the fundamental idea underlying most of our algorithms. So what are the inputs to our algorithm? This is a directed bipartite graph derived from the metabolic network, a set of seed metabolites, set of target metabolites, and an integer beta which tells you how big pathways you can generate and so on. And most importantly, what is the source set of metabolites with which we get the whole thing started? What is the food for the microbe so that it can produce whatever you are asking it to produce and so on. And we looked at a few examples. So you can see that we already started looking at very large networks. So we looked at a network of 3 organisms with about 14,000 nodes and 30,000 edges, and with as many as 1,000 metabolites that could be produced. And we looked at all pathways of size 20 to produce various metabolites and so on. So essentially, we were able to build community metabolic networks that are fairly large, and we are able to capture what kind of interaction goes on in this space. The next question is, how do we quantify these interactions? So we now found various pathways and so on, but how does one organism influence the other? And so does this help the organism, or does it kill the other organism, and things like that? And so to study this, we did a few experiments. So this was in collaboration with Professor Lars Blanc and Professor Smita from our department. So the question is, how well does an organism support another? And our approach was simple. We looked at what part of the metabolism of a single organism is enabled by the presence of a second organism, simply. So we came up with this index called Metabolic Support Index, which just quantifies what fraction of reactions that were originally stuck in an organism is now enabled in the presence of the second organism. And this is like an asymmetric index. So A might not support B much, but B might be very, very helpful to A. It's just how their metabolisms are wired. And if all reactions are stuck, there is no benefit, and the support index is 0. And the idea is to basically quantify what is the extent of help that one organism affords to another. And we had some experimental observations. We also did some experiments. The last row that you see here is our own experiment, where we saw that whenever the organism gets more help in the community, it grows at a faster rate and things like that. So this was, some simple validation of our approach, but as I'll tell you at the end, we are able to generate far more hypotheses than we can validate. So that's a bit of a challenge, but this paradigm is very useful for modeling very different systems. So we are able to answer many questions. So for instance, what are the metabolic capabilities of an individual organism or a community of organisms? What kind of metabolites are exchanged between these organisms? How do organisms depend on one another? Are they parasitic? Are they commensal? Are they mutualistically benefiting, and so on? And are there higher order dependencies? So how are these communities organized and things like that? And this has many implications for health and disease and so on. So one very interesting study we were a part of was, how does the gut microbiome recover post-antibiotic treatment? And if you see this cartoonish thing here, Very nice. It shows a forest fire, which is they like an antibiotic treatment to the gut to a forest fire, and then following a forest fire, the forest slowly recovers, the ecosystem slowly recovers, and similarly the gut also slowly recovers. And you'll be surprised that this takes as much as 12 months to a year, even 2 years post antibiotic treatment, and by that time you probably have had another dose of antibiotics. So it's a real challenge. But what they found was that there are a bunch of organisms, about 20 odd organisms, which are recovery associated, meaning they are able to, whoever had these organisms in their gut, their guts were able to recover much faster following antibiotic treatment. So, and why? What is so special about these 20 organisms? That is what we set to study. So, it turns out that they seem to have higher carbohydrate degrading capacity and so on. And there seems to be some specific synergy that was identified as well. And so, what we did was we did pairwise interactions between all pairs of these organisms. So we had 21 microbes, and we looked at pairwise combinations of these and built a microbial association network based on the interactions that we were predicting. And what we found was that the recovery associated bacteria were typically far more cooperative or helpful with each other than the others. So they worked together as a community, and they were able to produce potentially the various metabolites that are important for gut health and so on. You can also analyze these specific amino acids and things like other metabolites. So this was a network that we found, and this particular edge we thought was very interesting. And this was also verified in an experiment. So they had mice which were inoculated with these bacteria, and they were able to show a faster recovery of gut health compared to mice which did not have these bacteria and so on. So the point is that we still need to understand a lot more, but the interactions between various microbes seem to be driving gut health and so on. So we need to dig deeper into this. So what we did was we looked at a lot more organisms in the gut and tried to understand what kind of interactions are going on. I'm probably going to skip this because I want to get into the other study. So for example, in this study, we were able to look at what kind of metabolites are exchanged between the organisms. So there are some butyrate producers and some proteinate producers and so on, and there's a bit of lactate exchange going on between these organisms. All these short chain fatty acids are very important for gut health. So we were able to drill into some of these interactions and try to understand why some organisms are going to be more beneficial for the gut and things like that. So with that, let me get into a really interesting study. So this is the International Space Station, and it's obviously a clean room. So there are very few microbes there, but turns out that as humans go and as payloads arrive and so on, there are quite a few microbes, including important pathogens. And this is very important because the astronauts in space are typically immunocompromised, and I mean they have slightly lesser immunity levels and so on, and you want to make sure that they are not exposed to all these organisms. And I'd like to really acknowledge Venkat, Nitin and Chet, who are part of this study from NASA, and of course Sanjay and Rachita, who did most of the work here at IITM. So what did we do? There is a very nice data set that they've generated over years. So this is across time. So they collected several samples of microbes from the International Space Station in a gap of 2 months, and then after another 12 months and so on, and then identified all the viable microbes through some experimental techniques. But what we did was we looked at locations where there was a high presence of Klebsiella pneumoniae. This is the pneumonia causing organism, and looked at what organisms are supported by this organism, or how does this interact with other pathogens and things like that. And we had some very interesting studies. So we again performed this network-based analysis using our MedQuest approach, and also another methodology known as SteadyCom to understand what kind of are there parasitic interactions and things like that. So we studied both pairwise and community interactions, and we found very specific interactions between various microbes. So that is Klebsiella, which is a pathogen, but it supports other organisms called Pantavia and so on. And we found that there are many amensalistic and parasitic interactions. So in an amensalistic interaction, there is one organism that suffers a cost, and the other organism has no benefit at all. And in parasitic, both organisms, like there is one organism that suffers a cost and the other organism benefits. So these kinds of interactions we could identify in this very interesting microbiome. And what Venkat did was they also tried to experimentally study some of these predictions, right? So we found that a particular interaction between Klebsiella pneumoniae and an Aspergillus niger, and here, another related fungus was studied here, and what they found was that when the organism is by itself, there is no it grows very healthily, but in the presence of Klebsiella, the organism is like the growth is really hindered. So you see all this these nice structures are completely damaged in these images and so on. So it's just like some experimental validation of what we did, but there's obviously a lot more to be done. But to quickly summarize, we used this plain graph theoretic approach to study microbial interactions, and we also used a complementary constraint based approach. We also did some interesting studies here by trying to look at the organization of the community, because you typically have very similar organisms in these communities, and you cannot easily tease out what is their individual contribution to the community and so on. So we had some nice approaches to dig deeper into those. And we found that some of these pathogens, basically Klebsiella and Enterobacteriaceae, they are very important in mediating metabolic interactions. Not only they are a problem, they also help other organisms survive. So by potentially targeting Klebsiella in a targeted fashion, you might be able to disrupt a lot more organisms that are being found on the ISS. And we've generated a lot of hypotheses for experimental testing and potentially more insights into such clean room microbiomes, right? So you can also extend these observations to say an ICU or things like that, where you have very few microbes, but they still have like a very interesting network of interactions and so on. So really from outer space, we go into the deep sea, and the deep sea is really interesting because there is this portion near the ocean bed where you have very cold water mixing with very hot water. And what you find is in that temperature gradient, you have some really exotic microbes that live there, mostly extremophiles because they survive high salt, high temperature and things like that. But they have very interesting metabolisms, and we try to understand what kind of interactions happen between these microbes and so on. So this was with Karthik Raman in Wisconsin-Madison, his postdoc Jao, and of course my MS student Dinesh and our undergrad student Shreyansh. So we have applied similar approaches. Here I'm not going to go into all the details, but just tell you that these we found some very interesting microbes, even with like very reduced genomes that had interactions here. And one challenge with all these microbes is that you cannot grow them in the lab. In fact, you don't know how to grow them in the lab. So these experiments will probably suggest that maybe there are these communities or groups of microbes which can together grow. So you may be able to figure out how to even culture these in the lab and so on. And these microbiome data sets are very interesting because we think we've sequenced a lot of these microbes, but there is a whole lot of dark matter that's out there which we still don't know. And we do not, and it's very, very difficult to study them in the lab as well. But by doing such studies, trying to understand what are these interactions, we slowly start getting a picture of how these microbes are surviving, what they are doing, and so on. No, they just don't grow. We don't know what are the conditions required to make them grow. So some limitations. So the graph models just provide a static snapshot. So the very simple system, but it's very useful, but you still have only a static snapshot of the interactions. And well, as I said, some predictions agree with previous experiments, but we generate a lot more predictions than we can readily validate. And our algorithm obviously is sensitive to the quality of the input models and things like that. And we don't yet have any weights or rankings attached to the metabolites and paths. And as I said, we focus most of our analysis, in fact, all of our analysis on a metabolic view to the cell. But as I argued in the very beginning, it's a very important aspect of how cells interact. So that's still justifiable. But essentially, there's a lot more going on in the cell, so we need to see how to study these interactions from other perspectives as well. So I've showed you a few things today. So one is MetQuest, a tool to identify these metabolic exchanges and biosynthetic pathways. Metabolic support index, a metric to quantify the support an organism receives. Overall, this is a generic framework to understand microbial interactions and in essentially anything, right? So gut, it could be the gut, it could be the skin, or it could be a clean room microbiome and so on. So we've applied it to so many different scenarios to get insights. And there are a lot more interesting questions we can look at it. So these are some things we are working on right now. So what are the key differences between, say, pairs of microbiomes? So there's a person with diabetes, person without diabetes. How does their microbiome differ? Can we quantify the differences? Can we pin the health onto some combination of microbiomes? Okay, or in fact, now we are also thinking about looking at healthy and bleached corals. So coral microbiomes, again, is an increasingly interesting area of research. And how do we perform a differential analysis of how these microbes interact and so on? How do these communities assemble? What is the succession that happens there? What are the microbes that are keystone microbes, and what are the other microbes that come in and out? Are they very robust? Can these communities be invaded by new organisms? So many open questions there. And can we manipulate these microbiomes? Right? So there are people who say that you can have some curd and whatnot, and it will really manipulate your microbiome and make it healthier and so on. And I'm sure some of you who've taken antibiotics will get like a microbiome supplement these days, right? So how do you tailor that microbiome cocktail that makes your gut recover faster and things like that? And can you design minimal microbial communities for a purpose, right? So all these end up being very interesting mathematical optimization formulations and things like that. So if somebody wants to discuss later on. With it, I'd like to acknowledge a lot of people who were part of all of these studies. So with Aarti, Meghna, Rachita, Sanjay were involved in the ISS study. Niranjan was with the gut microbiome study. Venkat, Nitin and Shweta are from NASA. And Karthik, Dinesh, Amrita worked on, Karthik and Dinesh and Shreyansh worked on the Guaymas deep sea microbiome. And Amrita was also part of the ISS study. And I'd also like to acknowledge funding from RBCTSI and Unilever. Okay, with that, I'm happy to take questions. Thanks for your attention.